presentation, ladies and gentlemen. And we can proceed with the next presentation. So, ladies and gentlemen, boys and girls, MedTech stage in Riga at the Deep Tech Atelier conference, and our next speaker is Dr. Björn Heindryckx from Norway, and he'll be speaking about gene editing using CRISPR-Cas9 in the human germline. Is it a slippery slope towards designer babies? A very, very interesting topic. And a couple of words about Björn. He's Principal Investigator at Ghent-Fertility and Stem Cell Team, Department for Reproductive Medicine at Ghent University Hospital in Belgium, and also he holds a professor's position at Ghent University, and he's an author or co-author of almost 100 scientific publications. He's the co-founder of the Belgian Society for Stem Cell Research and also board member of the Belgian Society for Reproductive Medicine. Dr. Heindryckx, welcome. The floor is yours.
Thank you very much for your nice introduction. First of all, I would like to thank the organizing committee to give me this opportunity to present some of our research at your symposium. And I must say it was very professionally organized. I went to a lot of congresses, also online during the last year. And they were very helpful to me, especially also Kristaps. I just want to check with the technical staff. My clicking is still disabled to click my slides. Can you check this, if I can do myself my slides or not? Because for the moment it's disabled on my mobile phone. Otherwise, you will have to change my slides. So I will present this topic of gene editing using CRISPR-Cas. If it's a risk of slippery slope towards making designer babies. So gene editing in the human germline. Yes, I can now change my slides. Okay, so just a brief introduction. We are doing different kinds of research in our department. So I'm not working in Norway, I'm working in Belgium. My name, Björn, is a little bit Scandinavian name, but we are working in Belgium. So we have a busy IVF here in our clinic, helping couples to achieve a pregnancy. But our research is really focused on couples that we cannot still help with the current available techniques. So the upper part is some stem cell research. That's for patients who don't have sperm or oocytes that we want to make from stem cells, sperm or oocytes for them. It's not working yet, but in the long term, we hope we can resolve this. And then we have some more clinical translational research. Some topics are mentioned below. For example, this mitochondrial diseases is a very hot topic also in our field now. And we are trying to explore if this nuclear transfer technique by changing the nuclei, shifting the nuclei from a diseased oocyte to a healthy oocyte could resolve this. So with his experience in stem cells and embryology, we are now also started since 2 years this topic of gene editing, which is the topic of today. So gene editing in embryos and also in embryonic stem cells. So yeah, I would like to have visited Riga, but yeah, because of Corona times, it's very difficult to travel. I don't know if you were in Belgium. Most people know Belgium, only Brussels, but we work in Ghent. It's a much nicer city than Brussels, I would say. It's more historical. It's a student city. Myself, I'm from Bruges. It's a very small city, more in the west of Belgium. And it's also a very nice city. So if you have a chance, please visit these cities. instead of only Brussels. So I will briefly explain you the technology. I only have 20 minutes, so I have to be brief on this CRISPR-Cas system. I will present you some possible applications. So I will focus on gene editing in the human germline, so in oocytes, sperm, and embryos. So that's the human germline. And then I will end with some ethics and myths surrounding this topic and some new developments in the field. So you have to be aware, gene editing is not new. It's not something recent. Already in the late 70s, people have tried to manipulate the DNA to do some corrections or to modify the DNA. So there have been some techniques before CRISPR-Cas that have been used to change or modify the DNA. But since 2012, everyone is shifting to use this CRISPR-Cas system. So what is this CRISPR-Cas system? So it was originally detected in bacteria. So CRISPR stands for clustered regularly interspaced short palindromic repeats. It's not important that you remember this. So we will call it CRISPR system. So the CRISPR system and the Cas system, it's a system that you now can really accurately modify some DNA. So you can make a correction if a mutation is present in the DNA, or you can delete some genes to study the function of the genes. But originally it was detected in bacteria, and they used this CRISPR-Cas tool to destroy, when there is a virus coming into the bacteria, to destroy the viral DNA. So it's kind of mechanism to destroy the DNA, the viral DNA. So you have two important components. So you have this single guide RNA. So this is functioning as the GPS. So this will guide you. So that's why they name it single guide RNA. This will guide you to the specific position in the DNA that you want to manipulate. And then you have this purple thing, the Cas9 enzyme, who will make a cut in the double strand of the DNA. So when you make a cut with this CRISPR-Cas tool, so the single guide RNA will guide you to the specific position you want to manipulate. The Cas9 will be added to it, and it will make a double strand break in the DNA helix. And so when this double cut is made in the DNA helix, cells contain a mechanism to repair it. So you have this one system which is called non-homologous end joining repair. So it will insert some new bases, or it will make a deletion in the cut which was made. So you mostly use this left technique if you want to knock out the gene, to study the function of genes. But the cells also have this mechanism of another system called homology-directed repair. And in this system on the right, you add an extra piece of DNA that you want to build into the DNA. So this system is mostly used when you want to correct a certain mutation in the DNA. So the next slide, yeah. So you have the single guide RNA. It's working as a GPS to guide you in the DNA, in the position where you want to manipulate. And you have these scissors, the Cas9, the enzyme, who will make a cut. So there are systems available that you can design your single guide RNA easily. So there are online tools that will help you to design which single guide RNA you have to have. And this is based on the efficiency. So it will say you have to use this kind of RNA sequence, and this will be the most optimal, the most working efficiently. Because you see there, we have to minimize off-target effects. So we only want to manipulate in the position in the DNA that we want. We don't want that other cuts are made somewhere else in the DNA. So with these tools, you can really, predict a higher efficiency with these online tools that are available. the next slide. So yeah, CRISPR-Cas compared to other techniques, it's much more simple, it's much more efficient, and it's much more flexible. So that's why people are now using it. So we have to be aware, we have to check if the on-target efficiency. So when we want to manipulate or correct a certain mutation, when we do it using the CRISPR-Cas tool, afterwards we have to check if it has worked, of course, and we have to check for the second thing of side effects that nowhere else in the DNA that there have been changes introduced if you use this CRISPR-Cas system. A third thing that we should be aware of is mosaicism, especially in embryos. So we want to be sure that all the cells of the embryo are edited in the same way. So because some cells can be edited, some cells cannot be edited. So we have to be sure that all the cells, of course, are edited in the same way. But nowadays, technologies have advanced, especially in molecular techniques. So we can now use this next generation sequencing, mentioned the last. And this is working very accurately to check if our gene editing worked or not. So yeah, they found in the beginning some off-target things that happened and mutations that happened if you use this CRISPR-Cas tool in the beginning. But yeah, people are optimizing constantly the techniques. And now more and more we see that technique is working efficiently, especially in somatic cells and stem cells. But in embryos, we still don't know yet how efficient it is because it's still new, of course. So what can we do with this gene editing technology of CRISPR-Cas? So you can manipulate somatic cells, you can manipulate stem cells, but you can also manipulate embryos. And of course, if you manipulate embryos, this is more controversial because you will create finally an individual with all the cells manipulated. And most importantly, this edited cells will also be transmitted to the offspring. So that's why it's quite controversial to do it, of course, in the germline. So how we do, this slide just, they are also doing it on somatic cells. So if you see the left part of this graph, people are already using clinical studies using this CRISPR-Cas technology on somatic cells. So for example, this left person has some kind of disease. We take some patient cells from this person, this diseased person, for example, blood cells that we want to manipulate. So you can manipulate the blood cells in the lab, and then you can transfer back the blood into the bloodstream of the patient with some edited cells. But only cells in the blood will be edited, not in the sperm or in the oocytes of the patient. So it will stay in the patient. But what we do on the right side, we really manipulate on the embryo level. So it would be transmitted to the offspring, of course. So we can use it to correct certain mutations that lead to genes. We can now easily create knockout and knock-in mouse animal models. We can modify gene expression and so on. So there are a lot of applications possible with this CRISPR-Cas. And indeed, in somatic cells, it's already being used. The first study was in 2016. So it was in patients that had a terminal lung cancer. They could not do anything anymore. So they retrieved some stem cells from the blood from these patients. They did some CRISPR-Cas gene editing, and they put the blood back in the patient, making these blood cells more available to attack some, in this case, it was lung tumors. But we are doing it in embryos and stem cells. So if you want to know more, this is a nice review that we wrote with ESHRE a few years ago. So how you do it in embryos, so you need oocytes or fertilized oocytes, zygotes, and you just need to inject on the left side, it's seen, You just need to inject in the oocyte or in the fertilized oocyte your CRISPR-Cas tool, so the scissors and the GPS system, and for example, a template if you want to correct a certain mutation. And that's also done in human. So these are human pictures. So you inject with a needle your CRISPR-Cas tool together with the sperm, or you do it on the fertilized egg, or you can also do it on the embryo level. So once you have injected, you let the embryo grow. You see it in the upper part of the picture. And once you have blastocysts here in the middle, then you need to take some cells away from the blastocyst to see if the gene editing has worked, if the correction has been done, or if the knockout of a gene has been done. And you could also, because a blastocyst is not a lot of number of cells, you can also further create stem cells from this blastocyst. Then you have more cells to study and to see if the gene editing worked or not. So this mosaicism is a problem still in gene editing in embryos, because we see that some cells are edited, for example, but some cells are not. So that's not something we want. We want all the cells in an embryo modified in the same way. So we still need to resolve this. And I will explain you in the study in the next slide. There are now 10 to 15 studies in the human germline, so in embryos, oocytes, have been done. So not so much still. The first one was in 2015. We still don't have a lot of information of this gene editing in the germline. So for corrections, why we want to do this for corrections? So, you know, there are like 10,000 diseases that have a genetic origin. When they come to our clinic, we can do this technique called pre-implantation genetic testing. So we take some cells, as you see there, from a day 5 embryo, we take away some cells, and we check if the mutation is present or not. And the embryos that don't have the mutation, we transfer back to the patients. So we have a system that we can avoid transmission of genetic diseases. But of course, depending on the mutation or the deletion, a lot of the embryos will be affected. So only 25% will be available to get a healthy pregnancy. So with this CRISPR-Cas, we would be able to increase the number of embryos available to transfer back, because theoretically we can correct all the embryos and we could transfer back all healthy embryos to the patient. But there are some cases that we cannot do this genetic selection. So here you see some examples. For example, if both members, the first one, if both members of a couple are affected with the same monogenetic disease, for example, they both have cystic fibrosis, a mutation in a certain gene, all the embryos will be affected, and we cannot select it with this PGT technique. So for these couples, CRISPR-Cas gene editing would be a solution to still have a chance for their own genetic child. But there are some other examples written there. So people have tried it in research context, I must emphasize. This was the first important paper of the group of Mitalipov. So they injected some sperm who was mutated, who gave rise to some cardiac disease, and they checked if the CRISPR-Cas tool can correct this mutation. So they injected it into fertilized eggs in their first experiment. So you see here the syringe where they inject the CRISPR-Cas tool, and they saw that they could correct a number of embryos, that they could remove this mutation leading to cardiac disease. What they found was important, that they could avoid mosaicism if they injected a little bit earlier. So if you see in the left on the figure, if they inject the CRISPR-Cas tool together with the sperm, so already during fertilization, you could resolve this problem of mosaicism. So the earlier you inject this CRISPR-Cas tool, the better. This other study, I will not go into detail. You could also, knock out genes that are, as I said, because we still don't know in human embryo development which genes are really important for human embryo development. And that's, of course, important for infertility patients. So it's a very nice system to knock out a gene and to see the effects on embryo development. So we have done some studies, and also other people have done some studies to knock out the gene in an embryo and to see the effects on embryo development. So I will end with some ethics and myths around gene editing. Yeah, of course, it's not allowed in every country to do this gene editing. As I said, if you want to do it in embryos, you have to make embryos. And in a lot of countries, it's not allowed to make human embryos just for research purposes. Fortunately for us in Belgium, it is allowed. So we are with the happy ones. And also, yeah, some people, some countries have ratified this Oviedo Convention. And if you have ratified this, you cannot do any manipulation on the DNA. But we, Belgium, also didn't ratify this Oviedo Convention, so we are also with the lucky ones. In the beginning, it was actually the researchers that were afraid to do it in the human germline, like this Jennifer Doudna. She said we should not do it on embryos. And it was more the etici that were more favorable of doing it in human embryos. There are a lot of guidelines now, yeah, what we should do or not. And also with ESHRE, we wrote some guidelines. And with ESHRE, this European Society for Human Reproduction and Embryology, we are very much in favor of doing it, gene editing in the human embryos, but not in a clinical context. So only for research context to see if the efficiency is good and if it's safe and efficient to ever do in the future. Yeah, people are afraid that if you can do this, you will only, you will use it to make very intelligent babies, athletic babies, to choose the hair color. So this is the slippery slope, but we should not be afraid of it, because all our phenotypical traits, or hair color, or intelligence, or athletic ability, it's all orchestrated by a lot of genes that we still don't know and we still don't understand. So we should not actually be afraid that people will use it for making these designer babies, because simply we don't know how to do it, and we would have to manipulate a lot of genes at the same time, and this is at the moment at least not possible at all. Yeah, people are also doing it to get some attention. So like this guy, he doesn't have a medical problem, maybe except for his hair, but he's injecting CRISPR-Cas into his own body. But that's just to drive to get some attention to him. And of course, the CRISPR-Cas was a little bit in a bad context, because one guy already did it. So a Chinese professor already made children which were genetically modified. And that's a pity.
To jail, imprisoned, I guess, for several years for that, right?
Yes. He was sentenced for 3 years in prison. And I fully support this decision, because it was much too early, because we are still starting to get to know what's happening with this gene editing in embryos. But it was very much too early to do this already.
Björn, we are running out of time, actually. Sorry.
I will stop. There are some. I will end with one sentence. So the classical CRISPR-Cas technology, but in embryos, it seems that there are some problems. So we are now shifting more to new technologies. They are also CRISPR-Cas based, but they are newer technologies which might work more efficient. And in our lab, we actually do it because with our techniques of assisted reproductive technology, we know that we will make some children who also have fertility problems, because a lot of fertility problems are genetically caused. So we want to produce children who will not have infertility problems.
Yep.
So I will end with this slide, our research team, and I thank you for your attention.
Thank you so much, Björn. This is,
my pleasure.
I think this is the most interesting topic in the whole field of life sciences. And Dr. Heindryckx will be staying with us now virtually because this topic will be discussed a little more in detail. But before we start, ladies and gentlemen, another survey here on the MedTech stage. You can find the survey below the main window in your platforms. And again, there is one question and 4 possible answers. So please, let's start. The question sounds like that. Genetic engineering. Choose a word that describes your feelings the most. A, possibilities. B, scaring. C, responsibility.